Hello friends, welcome back to my channel, and today in this video we are going to study about frequently encountered signaling pathways. As because from the last two videos we are learning about the common steps and the overview of the signaling pathways, so I guess learning the signaling pathways will be easy for you. So this is the first pathway. What the pathway is saying is we already learned all these things, that these are TCR, that is T cell receptors, LCK, ITK, these are tyrosine phosphorylase that helps to phosphorylate tyrosine, and thereby they provide the binding site for the binding of this LAT, that is an adapter. Now adapter itself work as a docking site for the binding of other factors, enzymes and signaling molecules, like for here it helps to bind with PCLC gamma 1. Now what this PCLC gamma 1 does, it breaks PIP2 into DAG and IP3. IP3 have the receptor endoplasmic reticulum. With response to IP3, what endoplasmic reticulum do is it releases calcium. Calmodulin, which presents in the inactive state, binds with calcium and becomes active. How many calcium molecules bind with calmodulin for its activation? It is 4. As you can see here, 1, 2, 3 and 4. So 4 calcium molecules are needed to convert the inactive calmodulin into active calmodulin. Once it is activated, it binds with calcineurin. Calcineurin is a phosphatase. Phosphatase means it removes phosphate. So what it will do, it will remove phosphate from NF-80 and thereby making it active. It can easily cross the nuclear membrane now and can work as a transcription factor for the secretion of interleukin 2 that is responsible for T cell proliferation. So this is one pathway. Now the persons who are T cell mediated autoimmune, or in case of organ transplant, the drug used is cyclosporin. So what does cyclosporin do? It inhibits calcineurin. So once calcineurin is inhibited, NF-80 cannot become dephosphatase. The removal of phosphate from NF-80 won't occur, and because of which NF-80 cannot cross the nuclear membrane. So if it is not crossing the nuclear membrane, the gene won't get expressed. So that's how it prevents T cell proliferation. Now coming to the next pathway, this pathway starts from DAG. How the DAG produced? PLC gamma. We already learned in the last slide, from PLC gamma, it breaks PIP2 into DAG and IP3. So from IP3, one signaling pathway run that we learned in the last slide. Now from the DAG, what happens? With the DAG, this RAS is bound in the inactive form. The SOS works as a guanine exchange factor. It replaces GDP with GTP. So now with the RAS, GTP is bound. So in this form, the RAS is activated. The activated RAS will activate RAF. Activated RAF will activate MEK. MEK will activate ERK. How these are activating? With the help of kinase. RAS is phosphorylating RAF and making it active. RAF in turn phosphorylates MEK, converted into active. MEK in turn phosphorylates ERK and converts it into active form. Now this active form of ERK, it can cross the nuclear membrane and it will activate ELK. So once it activates ELK, so ELK and SRF together will activate a gene called FOS. The FOS will get phosphorylated and becomes active again. Now ERK can also directly phosphorylate FOS and activate it. So the ERK, it activates ELK. ELK and SRF together will synthesize FOS. And ERK, in other way, it activates the FOS formed due to ELK and SRF by phosphorylating it. Now with this phosphorylated FOS, its partner jun in the form of phosphorylation only, they both joined. Now this is an active jun-fos complex. This active jun-fos complex will activate AP1. AP1 is a transcription factor. So what it will do, it will bind to the DNA and work for the expression of interleukin 2, which is responsible for T cell proliferation. So once the receptor bind, so from IP3 also one signaling pathway runs. From DAG also one signaling pathway runs. From IP3 the pathway runs is the calcium signaling pathway, and from DAG the pathway runs is the Ras-Raf kinase pathway. And the next is from DAJ, one more pathway gets activated, that is of NF-kappa beta pathway. So with the DAG, PCAC theta will bind. It will activate TAC1 complex also and karma1 also. Now it's a cascade like karma1 will bind to BCL10, it will bind to Malti and then TRAF6. Now what TAC1 do? It phosphorylates IKK alpha. And what TRAF6 does? It adds ubiquitin to NEMO is a regulatory protein. And what it is regulating? It stops the functioning of IKK alpha and IKK beta. So TKA1 is phosphorylating IKK alpha, that is making it active, but it is still inactive because NEMO is attached to it. So TRF6, it degrades NEMO by ubiquitination. Now it is left with IKA alpha and IKK beta. Now what it will do? It will come and degrade I kappa beta. NF kappa beta and I kappa beta. They are in a joint form, and due to which NF kappa beta is in inactive form. Once it gets separated from I kappa beta, it becomes activated and can cross the nuclear membrane. And that's how this will act as a transcription factor again for the synthesis of internuclein 2, which is responsible for T cell proliferation.